So now we have completed, we're looking at the genetics booklet, but we've filled in all the way up through the dihybrid, up to what's called the dihybrid cross. So this is about the dihybrid cross right here. So, excuse me, that wasn't it. This is it. Okay. So here, a dihybrid cross involves two set of contrasting traits, so two different genes are followed. We could do a trihybrid cross where we're looking at three different traits at the same time, or a quadrahybrid cross. Now, the word hybrid is confusing here. When we went back and we were crossing, we were just looking at one trait like coat color here. This is called a monohybrid cross. But you might be saying to yourself, well, look at this. We have, this is not a hybrid. This is not heterozygous. This is a pure black and pure brown or whatever it was. So, but by convention, when we're talking about a Punnett square where we're just dealing with one trait and that has two alleles, we're calling that a monohybrid cross, even though there might not be a hybrid involved. And it has four squares in what's called the Punnett square. So in a dihybrid cross, we're going to look at di meaning two traits at the same time and see how they work. The key part here is this illustrates the principle of independent assortment, or also called independent orientation. What it means is that you can inherit one trait independently of the other. So we're going to look at color and whether your seeds are wrinkled or round. And what we're going to see is that you can inherit yellowness, for example, independent of roundness. You could be yellow and round, or you could be yellowed and wrinkled. Same thing with green. You could be green and wrinkled or green and round. And we're going to show how that works. So the symbols for a plant that has seeds that are pure yellow and pure round would be big Y, big Y, big R, big R. And we define what those mean over here. Pure green, which is the recessive, is little y, and little r, which is the wrinkled, which is also recessive. Hybrid yellow, or heterozygous, is big Y, and hybrid round is big R, little r. Okay, so this, for example, this plant looks exactly the same as this one in A. It's just that this one is carrying the other gene here, the other allele. So now what we're going to do is we're going to make we're going to cross a heterozygous yellow, and it should say heterozygous round seeded plant. So they're both heterozygous. Okay, so here's the genotype of one parent, which is the same as the genotype of the other parent here. And what you do is you have to make sure you have one Y and one R in each gamete. You would never have, unless it was an error, two Ys in the same gamete. So this right here is what we call, these are 2N. This is a 2N cell. It has two copies on their chromosomes. It has both homologous chromosomes in it. This one is the gamete. So this one is actually missing half the information. So this only has one of the chromosomes. So some gametes that could be produced by this cell are this, and this. These combinations of the possibilities. Same down here. So I put this one down here. These, and these. By convention, we start with putting the two capitals here. The two capitals here. It makes it easier to count. Then you go across and down, and you do the cross. So when you actually, this is representing fertilization here. Okay, so we did meiosis from here to here, and then we're doing now fertilization. So all of these are now diploid, and we got to look at what they look like, their phenotype ratio. Well, how many are yellow and round? If you count it up, this one's yellow and round, this one's yellow and round. So it's round, even though it carries that little recessive. This one's yellow and round. Yep, yellow and round, yellow and round. Not yellow and round. It's yellow, but it's green. Yellow, but it's wrinkled. This one is yellow and round, yes. And as we go through, you'll find out there are 9 of them. Then when you go through to yellow and wrinkled, to be wrinkled, you have to have the 2 little r's. Well, this one counts. This one counts. And what else? Where's the other one? Over here. So we have 3 of those. And then we count up the green round ones and the green wrinkled ones. And we show that this is a magical ratio you get when you cross 2 parents who are heterozygous for 2 traits with each other. You get 9 to 3 to 3 to 1. And then we can look at the ratios of just color. And you'll find you have 4 that are pure yellow, 8 that are hybrid yellow, and 4 that are pure green. And we can do the same thing for shape. We can say that there are 4 that are pure round, 8 hybrid round, and 4 pure wrinkled. So Over here, we've drawn to show you linked genes. Now, these are genes that are not linked. So they are not linked on the same chromosome. So if you get the yellowness, you're not necessarily getting round with it because they're not linked. They can separate during meiosis. But if they were linked, this would not work. So linked genes are going to mess you up. So if there's another gene over here for something and we try to do that, it's not going to come out with these ratios. All right, just to be aware of that. So according to the law of independent assortment, when two different traits are followed in a cross, they sort out independently when gametes form. So one trait does not depend on the other. So yellow aren't always round, green are not always wrinkled. But that only works, and this isn't written here, but it only works. You want to write that right over here if they're not linked. So it doesn't work for the linked genes. Independent assortment does not work for linked genes. Okay, how do I erase that? Clear screen. Okay, now we're not going to do this, but this right here is, I'm going to start you off with the floppy bunnies. I'm going to give you this one to do. So we have floppy-eared bunnies. In rabbits, black hair is dominant to brown hair, and long straight ears are dominant to floppy ears. And also black nose, which we should have written here, is dominant to pink nose. Okay. So we made all these letters. A male rabbit with the genotype big B, little e, is crossed with a female rabbit with the genotype little b, big E, little e. Okay. So he would have a black nose, this one, and he would have floppy ears. And this one would have a pink nose and have long ears. But you have to first make the gametes. So from this one right here, if you do meiosis, the only gametes this organism can make are big B, little e. So this would all be big B, little e across the top of here, from here. This is what's coming from here, doing meiosis. Now over here, this one can make either big B, little e, because the little b can go with the big E, sorry, or it can make little b, little e over here. I can't talk. So here we go. Now, there are shortcuts to this. You don't have to, like, for example, you might be saying to yourself, I'm good at math. I don't need to do this, this, and this. This is just going to be, if I do one of the column, I can just do it across. So sometimes I will do that. I'll say this, and I'll just draw an arrow to say it's all across. And then I'll say whatever I got here is just all across, and I'll write that like that. But for the first one, it's good to do it out just to get the idea. And so what you find out of 16 is that you end up with this 8 to 8 to 0 to 0 ratio of the phenotypes. And you could even reduce that to 1 to 1 to 0 to 0 as well. If you on a quiz, that ratio of 8 to 8 to 0 to 0 or 1 to 1 to 0 to 0 would be equivalent. All right, so that's one I'm going to have you do. And so that's the dihybrid cross. Oh, another thing to know is that you can do a dihybrid cross, and it doesn't have to be with two things that are dihybrid, like we just saw. They're not dihybrid. They're not actually hybrid for both traits. And so that's why we didn't get that 9 to 3 to 3 to 1 ratio. We got a different ratio because the gametes were different. We still call it a dihybrid cross. Okay, thank you very much.